In this video, we are going to talk about biotransformation or metabolism. So what is biotransformation? It means chemical alteration of the drug in the body. It is needed to render nonpolar or lipid-soluble compounds to polar compounds or lipid-insoluble or water-soluble compounds, so that they can be excreted easily. So that it can be easily excreted and not reabsorbed in the renal tubules. So basically, lipid-soluble substances are transformed to water-soluble substances. If we look at hydrophilic drugs such as streptomycin, neostigmine, etc., they are already hydrophilic, so they are largely excreted unchanged in urine. Little biotransformed and they are excreted easily because they are already hydrophilic. Now, the primary site for drug metabolism is liver, okay? And the others are kidney, intestine, lungs, and plasma. The primary site is basically liver. Now, the biotransformation of a drug may lead to any of the following, which includes inactivation, active metabolite from an active drug, or activation of inactive drug. This depends on the drugs. So first, let's see inactivation. Most of the drugs and their active metabolites are rendered inactive or less active. Examples of the drug include ibuprofen, paracetamol, lidocaine. So the next one is active metabolite from an active drug. So many drugs are converted to active metabolites as well. Let's take a look at some of the examples. Here we have the active drug and the active metabolite on the other side. First one is chloral hydrate. It is converted to the active metabolite trichloroethanol. Morphine is converted to morphine 6-glucuronide. Allopurinol is converted to alloxanthine. Diazepam to oxazepam, digitoxin to digoxin. So these are a few of the examples whereby the active drug is converted to active metabolite, and this happens in biotransformation. Now the third one is activation of inactive drug, or we can simply call it a prodrug. Such drugs are called prodrugs. That is, they are already inactive and they needed to be converted to one or more active metabolites. Okay. What are the advantages of this prodrug? They are more stable, they have better bioavailability, and they have less side effects and toxicity. So they offer advantage over the active form. Now let's take a look at some of the examples of prodrugs which are converted to active form inside our body. So here we have prodrug on this side and active form here. First drug is levodopa. It is converted to dopamine inside the body, which is the active form. Enalapril to enalaprilat, dipivefrin to epinephrine. Proguanil is converted to the active form cycloguanil. Prednisone to prednisolone. So these are some of the examples of prodrugs and their active forms. Now let's take a look at the biotransformation reactions. So here you can see they can be mainly classified into non-synthetic or phase 1 or functionalization reactions and the second type is synthetic or phase 2 or conjugation reactions. So these are basically how biotransformation reactions can be classified as. Now let's take a look at some of the examples of these reactions. First one in phase 1 reaction is oxidation. Oxidation is the most important reaction, and it involves the addition of oxygen or negatively charged radical, or we can say that it is the removal of hydrogen or positively charged radical. And it is usually carried out by monooxygenase enzymes in the liver. Next one is reduction. As you know, it is the opposite of oxidation, and it involves cytochrome P450 enzymes. Alcohols, aldehydes, quinones are reduced. And the examples of drugs which are reduced include chloral hydrate, chloramphenicol, halothane, warfarin, etc. Now the next phase 1 reaction is hydrolysis. It is the cleavage of drug molecule taking up a molecule of water. It occurs in the presence of esterase enzyme, and the examples of drugs include choline esters, procaine, lidocaine, procainamide, aspirin, carbamazepine epoxide, etc. And hydrolysis mainly occurs in liver, intestines, plasma, and other tissues. Now we will obtain a polar metabolite from the phase 1 reaction. And now what happens in the phase 2 reaction is that if the metabolite from phase 1 metabolism is sufficiently polar, it can be excreted by kidneys. However, many phase 1 metabolites are too lipophilic to be retained in the kidney tubules. So this is where phase 2 reactions come into play. Here, a subsequent conjugation reaction with an endogenous substrate, such as glucuronic acid, sulfuric acid, acetic acid, or an amino acid, results in polar, usually more water-soluble compounds that are most often therapeutically inactive. So we get a more polar compound at the end of phase 2 reactions. Now let's take a look at what are the phase 2 reactions or the synthetic reactions. First one is glucuronide conjugation. Glucuronide conjugation is one of the most important synthetic reactions and is carried out by a group of U2P glucuronyl transferases or UGTs. U2P glucuronyl transferases are the enzyme which is used in glucuronide conjugation. Compounds with a hydroxyl or carboxylic acid group are easily conjugated with glucuronic acid, which is derived from glucose. Examples of drugs include chloramphenicol, aspirin, paracetamol, morphine, etc. Not only drugs, you see, endogenous substances like bilirubin, steroidal hormones, and thyroxine utilize this pathway of glucuronide conjugation. Glucuronidation increases the molecular weight of the drug, which favors its excretion in bile. Next one is acetylation. Compounds having amino or hydrazine residues are conjugated with the help of acetyl coenzyme A or acetyl-CoA. Okay, so this is the enzyme used here. Examples: sulfonamides, isoniazid, hydrolysine, clonazepam. Next one is methylation. The amines and phenols can be methylated. Examples of drugs include adrenaline, histamine, nicotinic acid, methyl dopa, etc. Okay, so the next one is sulfate conjugation. The phenolic compounds and steroids are sulfated by sulfotransferases, or we can say SULTs. Sulfotransferases. Examples of drugs conjugated by this method include chloramphenicol, methyl-dopa, etc. And next one is glutathione conjugation. As you know, this usually occurs in the case of paracetamol in order to prevent toxicity. Okay, so that's all about the biotransformation reactions and what is biotransformation in brief. Hope you liked the video.